For over 160 years, science had the origin of snakes completely wrong. Not partially wrong, not slightly off. The family tree was built on a misidentified bone, a single cheekbone, the jugal, incorrectly labeled in fossil specimens since the 1860s. Every phylogenetic analysis, every evolutionary timeline, every textbook diagram descended from that one error. And because that bone was wrong, the geographic origin was wrong, the ancestor was wrong, and the entire origin story that paleontologists had been teaching for generations was wrong. The correction came from Argentina in 2019, from a 95-million-year-old skull so well preserved that the internal nerve canals were still intact inside the rock. When researchers ran micro-CT scans through that specimen and corrected the jugal identification, the snake family tree reorganized entirely. What replaced the old story is more precise, more specific, and considerably more strange. Before the origin can be understood, the anatomy demands attention, because the snake body is not simply a lizard without legs. It is a structural departure so extreme that it requires dedicated explanation. A reticulated python carries between 300 and 400 vertebrae. A human carries 33. The Hox genes, the master developmental switches that build vertebrate bodies, are present in every living snake, unchanged. They do not instruct a different body plan. They instruct the same plan, then stop completing specific steps. The limb program initiates. The signal is sent through the same molecular pathway that builds arms in every other vertebrate on the planet, and then it stops. Not because the instruction is missing, but because a downstream door no longer opens. The skull is equally modified. 7 independently mobile bones allow a snake's jaws to expand beyond 150 degrees, enabling prey consumption wider than the snake's own head. This is not a simple hinge. It is a controlled, sequential expansion of 7 separate skeletal elements, each operating on its own mechanical schedule. And the numbers are significant. Approximately 3,950 living species, an estimated 3 billion individual animals, present on every continent except Antarctica, in habitats ranging from deep ocean trenches to alpine elevations above 4,000 meters. By species count and individual count, snakes are among the most successful vertebrate experiments in the history of life. Where this body plan came from and when was, until recently, genuinely unknown. The dominant hypothesis for most of the 20th century placed snake origins in the ancient Tethys Sea, the shallow, warm body of water that once separated Laurasia in the north from Gondwana in the south, spanning roughly where the Mediterranean and Middle East now sit. The logic was as follows. The oldest well-preserved fossil snakes with visible body plans, Paterhotchs and Eupodopus, legged snakes from limestone deposits in Lebanon and Israel, dating to approximately 95 to 100 million years ago, were found in marine rock, marine fossils, therefore marine origin. Early molecular studies pointed toward a close relationship between snakes and varanid monitor lizards, a group native to the Old World, adapted to warm coastal environments in Africa and Asia. The geography aligned. The morphology seemed to align. The hypothesis was coherent. Then, in 2015, it appeared to be confirmed entirely. A fossil in a British museum, labeled as an unidentified vertebrate and overlooked for years, was identified by paleontologist David Martill of the University of Portsmouth as the first 4-legged snake ever recorded. 4 legs, snake anatomy, a missing link. Specimen was named Tetrapodophis amplectus. The scientific community responded with considerable excitement. The aquatic origin, the Old World ancestry, it all appeared to be confirmed. The debate, most researchers assumed, was settled. It was not settled. Within years, the Tetrapodophis identification came apart. Michael Caldwell, vertebrate paleontologist at the University of Alberta, was skeptical from the beginning, but access to the specimen was blocked. The fossil had been removed from Brazil without the required export permits, placed in a private collection in Germany, and independent examination was effectively impossible for years. When Caldwell and his colleague Robert Rice of the University of Toronto, Mississauga were finally permitted to examine the specimen directly, their findings were unambiguous. Published in the Journal of Systematic Paleontology in 2021, their analysis showed the following. The skull lacked the loosely articulated bones that define snake jaw structure. The tooth arrangement was wrong. The vertebral morphology was wrong. The animal was almost certainly a dolichosaur, an extinct group of elongate marine lizards from the Cretaceous period, not remotely related to snakes. The 4-legged snake that had resolved the origin debate was not a snake. But the deeper problem was not one misidentified fossil. The deeper problem was that snake origins research had been conducted for over a century with fundamentally incomplete material. Snake skulls are highly modified, built from loosely articulated bones that scatter rapidly after death. Almost no complete Mesozoic snake skull had ever been recovered. Researchers were constructing evolutionary trees from isolated vertebrae, jaw fragments, and partial impressions, and the foundational anatomical identifications embedded in those analyses had never been rigorously challenged. The jugal bone, present in fossil snake relatives since the 1860s in the literature, had been consistently misidentified by generation after generation of researchers, not because of negligence, because no specimen existed with sufficient preservation to force a correction. Until Najash. In February 2013, Fernando Garbarolio, an undergraduate paleontology student at Universidad Maimonides in Buenos Aires, was on his first field expedition at a site called La Buitrera, the Vulture Place, in the Rio Negro province of northern Patagonia, Argentina. He was accompanied by his supervisors, Sebastiana Pestaguilla and Guillermo Ruggier of the University of Louisville. Garbarolio found a skull. It was, as the subsequent analysis confirmed, the most complete Mesozoic snake skull ever recovered. The specimen was approximately 95 million years old, preserved in fine-grained sandstone from a Cretaceous environment, a semi-arid landscape of dune fields and ephemeral water bodies. The rock quality was exceptional. Internal nerve canals, vascular passages, and the precise three-dimensional relationships between skull bones were intact inside the stone. The team applied microcomputed tomography, the same scanning technology used in medical imaging, capable of reconstructing internal anatomy in 3 dimensions without physically cutting the specimen, to map the skull's internal structure. The animal was named Najash Rio Negrina. Its genus derives from the Hebrew Nahash, the legged serpent of the Book of Genesis. Its species name honors the province. It was 95 million years old. It had functional hind limbs, and its jugal bone, now clearly visible in the CT reconstruction, was in entirely the wrong position relative to where a century of literature had placed it. The correction, published in the journal Science Advances in 2019, reorganized the snake family tree. When the jugal was correctly positioned and the phylogenetic analysis rebuilt with accurate anatomy, the results were specific. Najash and related South American and African Gondwana lineages moved to the base of the snake family tree, confirmed as the deepest diverging snakes known outside the crown group that gave rise to every living species today. The marine Lebanese and Israeli snakes, Pachyrhachis and Eupodopus, were repositioned as specialized late diverging offshoots, not primitive ancestors. The Tethys Sea hypothesis was displaced. The first confirmed snakes did not come from the sea. They came from land, specifically from the warm, semi-arid interior of Gondwana, in what is now Patagonia, Argentina. The confirmed age of the snake lineage itself also extends substantially further back. In 2015, Caldwell and colleagues published 4 new snake specimens in Nature Communications from Jurassic and early Cretaceous localities in England, Portugal, the United States, and Morocco. The oldest pushed the confirmed snake origin to approximately 167 million years ago, the Middle Jurassic period. The snake lineage was already ancient when the first flowering plants appeared. It predates Tyrannosaurus rex by 90 million years. One correction to one bone, and the entire established origin story was wrong in its geography, its ancestry, and its environment. The why of snake origins is now better understood than the where, and it is ecologically specific. During the period, paleontologists designate the Cretaceous terrestrial revolution, approximately 125 to 80 million years ago. The land surface of Gondwana underwent a transformation. Flowering plants radiated rapidly across continental interiors. Insect diversity exploded in response to new food sources and habitats, and small burrowing mammals began occupying underground environments in substantial numbers for the first time. The prey went underground. A surface-dwelling lizard lineage in Gondwana found itself at an ecological threshold. In tunnel systems and burrow networks, forelimbs were mechanically disadvantageous. They increased the turning radius required to navigate confined passages and created resistance during lateral movement. A body capable of compressing to any cross-section, moving without legs swing arc, and striking in confined space without preparatory room. That body had access to food that four-limbed competitors could not reach. The 2023 large-scale snake genome study published in Cell, analyzing 14 high-quality snake genomes across all major living lineages, identified the specific genetic events underlying this transition. The gene RDH10, which initiates four-limb development in vertebrates, is absent from the snake lineage. The gene HOXD12, critical for limb patterning in every other tetrapod, is absent from all 14 genomes analyzed. These are not gradual reductions. They are specific gene deletions. The genome removing instructions it selected against over millions of generations. The trunk elongated through restructuring of the HOX gene regulatory landscape, multiplying vertebral count from the ancestral tetrapod range of approximately 30 to the 300 or 400 seen in living pythons. The skull reorganized into its 7-bone flexible structure. And approximately 100 million years after limb loss, venom appeared, not as the founding innovation of snake success, but as a later refinement layered onto a body plan that had already survived on 4 continents and through at least one mass extinction. Venom is not what made snakes successful. It is what made successful snakes more efficient. The Cretaceous-Paleogene mass extinction event, the Chicxulub asteroid impact 66 million years ago, destroyed approximately 83% of all squamate species on Earth, including every member of the giant Gondwanan snake lineages that had dominated the southern continents for over 30 million years. Madsoya, from Patagonia, and Gigantophis garstinici, from Eocene Egypt, had reached estimated body lengths of 9 to 10 meters. They were among the largest snakes that have ever lived. They radiated across South America, Africa, and southern Europe for tens of millions of years. The asteroid impact eliminated them entirely. Not one living snake is descended from them. What survived were small-bodied generalists, burrowing species and insectivores capable of sheltering underground, with low metabolic requirements sufficient to persist through the impact winter. A 2021 study by Catherine Klein, Nicholas Longrich, and colleagues published in Nature Communications combined molecular clock analysis with fossil calibration data to map the timing of post-extinction snake diversification. The result was unambiguous. The explosive radiation of crowned snakes, the event that produced cobras, vipers, pythons, mambas, rattlesnakes, sea snakes, and the remainder of the living fauna, was triggered directly by the Chicxulub impact, emptied ecological niches, eliminated competition, and cleared continental land masses, created the conditions for rapid diversification. The major clade Aphrophytia colonized Asia in the early Paleogene, spreading across the post-extinction world. Modern snakes are the direct product of that catastrophe. The asteroid that ended the Mesozoic did not simply kill the dinosaurs. It created the conditions under which the snake world as it exists today became possible. And the giant snake body plan, the 10-meter predator body, evolved a second time independently in the anacondas and pythons of the modern era, 2 separate evolutionary lineages, tens of millions of years apart, producing nearly identical body sizes through entirely distinct genetic pathways. The genome distinguishes them. The eye cannot. Every living python and boa carries visible physical evidence of this evolutionary history. At the base of the tail, on either side of the cloaca, a small claw-like structure protrudes from the scales. These are pelvic spurs, the external remnants of hind limbs. Beneath them, inside the body, a vestigial pelvic girdle still forms during embryonic development. Bones present, partially developed, reaching towards a limb structure that will not be completed. The 2023 Cell Genome Study confirmed that the developmental program for hind limb construction is still running in these animals. The genetic instruction proceeds through multiple stages. At a specific point in the developmental cascade, the program is interrupted, not by the absence of the gene, but by the absence of downstream regulatory elements that would complete it. The snake is not done with its own evolution. The body plan remains in transition, 95 million years after Najash walked on functional hind legs through the Patagonian dunes. On the question of venom, the 2013 King Cobra Genome Study published in the Proceedings of the National Academy of Sciences, led by Freek Vonk and colleagues, traced the origin of venom apparatus genes. Snake toxins were not novel genetic inventions. They were co-opted, recruited from pre-existing genes already functioning in the animal's body. The venom gland shares evolutionary origin with the pancreas. The toxins derive from digestive enzymes, the most pharmacologically complex biological weapon produced by any vertebrate on Earth is a repurposed digestive system redirected outward. Evolution does not build new tools from nothing, it modifies existing tools. This principle, the repurposing of existing genetic machinery, runs through every major innovation in the snake body plan. The snake did not merely coexist with early primates, it shaped them neurologically. Research published by Lynn Isbell, professor of anthropology at the University of California, Davis, in her 2006 paper in the Journal of Human Evolution, and expanded in her 2009 book, documents dedicated neural pathways in the primate visual cortex specifically tuned to snake detection. The pulvinar nucleus, a structure in the primate thalamus, shows faster and stronger activation responses to snake-shaped stimuli than to any other. Visual threat category tested, including spiders, faces, and geometric shapes. These responses are present in primates with no prior snake exposure, indicating a genetic rather than learned origin. The argument is straightforward. The primate visual system underwent selective pressure from snake predation over tens of millions of years. The snakes that existed during primate evolution, already 100 million years into their own evolutionary history, already equipped with flexible skulls and chemosensory detection, were among the most consistent predation pressures early primates faced. The result is a dedicated detection system, wired into the primate brain at the architectural level. This reframes the relationship. Every human being alive today carries a neurological system whose design was partly authored by snake predation pressure. The alertness, the rapid visual scanning response, the instinctive recoil, these are not cultural. They are the physical record of a predator-prey relationship that ran for tens of millions of years. The snake family tree stretches back 167 million years. The human fear response to snakes is, in the most literal biological sense, a tribute to how effective that body plan has been. The snake genome studies of the last decade have not closed this field. They have opened it. The 2023 Cell study identified, for the first time, the specific regulatory elements controlling the transition zone between neck and trunk in snakes, the molecular boundary that determines how many vertebrae are dedicated to swallowing versus locomotion. This boundary varies significantly across lineages and appears to have shifted multiple times in snake evolutionary history, enabling different prey specializations. The mechanism is now identifiable at the base pair level. Separately, comparative genomics has begun mapping the precise sequence of gene loss events in snake evolution, constructing a molecular timeline of when forelimbs vanished, when the skull reorganized, when the chemosensory system expanded, and when venom first appeared. These timelines do not always match the fossil record. Where they diverge, the divergences identify gaps in the fossil record, periods and places where snake fossils have not yet been found but must exist. Northern Gondwana, what is now northern Africa and southern South America, contains the predicted geological formations. The fossils are there. They have not been excavated. The origin story of the snake is not finished. It is an active research program running simultaneously in genomics laboratories, CT scanning facilities, and field sites across South America, Africa, and the Middle East. Each year produces corrections to the previous year's conclusions. The snake lineage spent 167 million years becoming what it is. The science of understanding it is considerably younger and considerably less complete than it appeared even 10 years ago. If this kind of research, the real science with the specific findings and the named researchers, is what you want more of, subscribe. Every week covers a different origin story that the standard version gets wrong. The next one is already in production.